Without further ado, I'd like to introduce our guest speaker for tonight. Ramesh Sagili is an assistant professor in the Department of Horticulture and the Oregon State University Extension Service. And he'll talk about the current status of honeybees in Oregon and the United States and the crops that they pollinate. And Dr. Sagili came to Oregon 2 years ago, right? 2009, from Texas, which bears a little similarity to his native land, South India.
Yeah.
A little warmer than Corvallis, a little drier. But
we see the sun a lot. So.
You see the sun more than here. But, you know, the sun is always here. You just have to go to the right place to find it.
That's right.
And he got his Master's of Science in Agriculture from the Andhra Pradesh Agriculture University in India and his PhD from Texas A&M. So thank you for being here. We're glad you're here.
Yeah, thank you. Good evening, and can you all hear me well in the back as well? Can you hear me? Perfect. How about my accent? Is that good? Yeah, I always joke, my accent keeps people awake, so that's fine. So you'll not be sleeping during my talk. That's good for me. So yeah, thanks for inviting me, Nick, and I think it's a very important topic, and that's why I see a lot of audience here today, and I'm excited to be here as well. And thank all of you for coming. And the topic, actually, Nick suggested me this topic. This is not my title. So bees on the brink. So I'll go with that title. I like that. So I'll talk a little bit about, before that, I just wanted to know how many of you, like I saw here, he got a prize. He just got one wrong here. So he has some beekeeping experience, I can say for sure. But how many of you have bees in their backyard or had bees before? Okay, there are at least maybe 5% for sure. Okay, and then how many of you have looked inside a hive? Like, have you seen a queen and a bees inside the hive as well? Okay, there are quite a bit. If you haven't, then we have an observation hive here. There is a queen as well in this, and then you can, after the event, maybe you can come here, and when the lights are on, you can see the hive as well here. So this is called an observation hive. So yeah, I'm asking because I wanted to pace the talk based on how much biology I should talk. So I'll give a brief introduction on importance of bees, and then I'll go a little bit on biology, and then mostly I'll talk about the problems or the challenges that the honeybees are going through for the last 5 years at least. And then I'll talk a little bit of what you can do as citizens or backyard beekeepers, or even as a person having some apple or some other kind of tree on your backyard as well. So we'll start with that, and I always like to put this slide in front, you know, and I think if you had already gone through this slide, you would have answered most of your quiz questions as well, I guess, because some of the answers are here on this. So the honeybees, all bees are very important, but I'm just talking about honeybees here. So bees are important not only in economic terms because they pollinate about crops worth $20 billion in the United States alone, but also because they are also important for ecosystem. Even the wild plants which are reproducing are also dependent on bees for their pollination and reproduction and growth. So bees are not only important for our crops that we grow, but also for our wild crops that are in the wild. So in Oregon, if you ask me, it's about $500 million worth of pollination that the honeybees do. And there are 90 different crops that are being pollinated by honeybees alone in the United States. And for those who are interested in some statistics, there were about 2.5 million hives according to this 2007 study. And if you want a figure, 60 years ago, we had about 5 million managed honeybee colonies. When I say these numbers, these are all managed honeybee hives. At this point, we don't have much. It's only like 1% of feral or wild hives that are there in the wild. But most of them, 99%, are the managed hives that you see by beekeepers. So this number has declined by about 60%. There are several reasons for that. If you have some beekeeping experience and have been reading the news, sorry, I just switched to the last one, I guess. So there are several things from Varroa mite, which you guys answered. A big threat the beekeeping industry has been going through for the last 20 years or more. And then there is several other reasons. It's not a lucrative business. We think beekeeping might be there. The beekeepers make a lot of money by just producing honey or pollinating. No, actually they are in trouble. Beekeeping is not a lucrative industry or a lucrative business. That's the reason you don't see too many new beekeepers getting into that industry as well, to keep bees and make money. So it's kind of troublesome. So about 1/3 of our diet literally comes from bee pollination. We talked about that in the quiz as well. Just wanted to put this picture to show how important they are in Europe as well. So I just stole this picture from a book. So honeybees are here somewhere on top of cattle in the pork industry. So they're very important, valuable domestic animals in Europe as well. And I always put this slide as well. I have several fruits missing here that are being pollinated. This is whatever I got from the Safeway to get this picture. So there are several things which are missing. So these are all yummy stuff that you see. All the healthy stuff is dependent on honeybees for pollination. And then just imagine that you take honeybees out of the ecosystem, then you will end up something eating like that. I know it's a bit of an exaggeration, but still that's pretty much what I can say. So you have you seen anything like this before? Okay, so these are honeybee hives. These are commercial bees the beekeepers have put. So each of these you see here, these are 1, 2, 3, 4 hives on each pallet here. So these are like thousands and thousands of hives. So this is a picture that was taken when the beekeepers go down there in California, as Nick was mentioning. Almonds are basically in California, the San Joaquin Valley or the Central Valley. So they are the huge, they need about 1.5 million hives to pollinate almond crop. So without honeybees, there is no almonds that you eat. And United States, especially California alone, is the largest producer of almonds in the world. So almost 100% of our supply comes from California. And without bees, there is no almonds. So this is a picture where you see all these hives. This is called a holding yard. So the beekeepers have to hold their bees here before they go into orchards for pollination. So once the orchard is ready with 5% or 10% bloom, or maybe even before that, then the farmer asks the beekeepers to get the bees into their hive, and they put 3 or 2 per acre, and then they go for pollination from there. So it's a huge industry as well, which we don't know. We usually talk about bees means just honey. No, they do a more of an important job as pollinators rather than just producing honey. So here are some crops I have listed in Oregon that are being pollinated. These are just major ones. There may be missing some as well. Blueberry, cherries, pear, apple, caneberries, cranberries, strawberries, clovers, all kinds. Meadowfoam. Have you heard of meadowfoam? Have you seen meadowfoam crop before? Yeah, I see some of the heads nodding. So meadowfoam is another, it's a oilseed crop. It just grows maybe a feet tall, I guess. And it's all white. If you see, these are the hives in between that crop. So it's all white, and you see this in Harrisburg area. If you're going on I-5, if you pay some attention, you'll see some on your right and left as well. So this is a crop which even for me, it was new moving from Texas. I never saw a meadowfoam crop before. So this is also dependent on honeybees. So several crops are there which are dependent on honeybees for pollination. And several vegetable seeds like carrot, onion, they're all dependent on bees as well. And as you know, since last couple of years, we have seen bees in news, and because of all this colony collapse disorder. And here you see a honeybee hive in the White House, and I heard they produced a lot of honey last year, so they gave it to some foreign dignitaries as well. So they produced at least maybe 100 pounds or more, I guess. So for those who haven't looked at a beehive or don't know much of biology, I just wanted to start a little bit of biology here, and because honeybee biology is so fascinating. I can talk for 365 days and still will be left with something else. But I'll just quickly, within the next 1 or 2 minutes, I'll cover this thing. So they're called eusocial insects. So for those who have some entomology background, a eusocial insect has to satisfy 3 different things. There should be overlapping generations. There should be a reproductive division of labor. That means the queen is the only one that lays eggs in the hive. And there is cooperative brood rearing. That means all the bees together, they take care of the larva inside the hive. So that's basically, those are 3 things that honeybees have to satisfy to be called a eusocial insect. And they have a very well-defined caste system with 3 different individuals here. Queen, which is the mother of all the individuals in the hive. And she's a diploid organism. That means she has 2 sets of chromosomes. Then workers are also diploid, having 2 sets. And we call them facultatively sterile. That means they have the ability to lay eggs. Workers can lay eggs as well. But those eggs will become drones or males because they don't have the mating apparatus. They don't have the spermatheca to store the sperm. So they are laying only the drones. So it's a haploid one they're laying. So the drones are the males, and they're haploid, 1 set of chromosomes. And depending on the time of the year, you may see maybe a few hundred drones in a hive. Basically from summer and spring on, you will see most of the drones being reared in a hive. and workers are about thousands of it. If you ask me a standard hive, 2 boxes, maybe there are 30,000 to 40,000 bees in there. So this is basically about it, and this is one of our experiments that I stole, and here is the queen, and always you'll see the bees surrounding her. They are either antenating her, licking the pheromone from her, and also feeding. The queen doesn't feed by herself most of the time, so the workers are providing her the proteinaceous secretions, or worker jelly, or royal jelly, what we call. So they are producing the jelly and feeding the queen as well. So at least one person in each of the talks that you asked me, oh, it's easy to find a queen because queen is born pink. But basically, this is not the case. We paint the queens pink for our purposes so that we can find the queen inside a hive. So just don't be in the... So this is morphological differences. You can see queen is oblong. You can see a queen here as well in this observation hive. So they have a tapered body, elongated body, and then the drones are little fat, barrel-shaped with bigger eyes, and the workers are the smaller ones you see. Can we get to that? So I just put a video clip for those who haven't seen inside a hive. So here you see a queen, and she's being fed by this worker bee here, and these bees are kind of licking her or antenating. So there is a pheromone the queen releases. So that pheromone is very important for the integrity of the hive. If you lose a queen, I mean, within 4 hours, they'll be able to find out that there is no queen, and they start making a new queen. So that's a very fascinating biology they have. So now the queen is looking at, we call it a patrolling behavior. She's looking for a cell which is clean so that she can put her back or the abdomen and lay an egg. So once she finds one clean cell, she may just put her abdomen and lay an egg. Now she's laying an egg here. It takes about 8 to 10 seconds to lay a single egg, and queens can lay about 2,000 to 2,500 eggs in summer and springtime. So they're pretty productive in laying eggs.
No privacy, right?
No privacy, yeah. And this here I have a worker behavior. I just wanted to show what is that cleaning behavior. So we put the tags on top as well so that you don't miss the bee. We paint their backs as well. So you see the paint on the back so that, because they are all same, if you just miss, you can't record the same bee. You may think this is the bee which you were recording before. So now she has put her head inside the cell for more than 3 seconds. So we call typically it's a cleaning behavior. She's cleaning with the mandibles so that the queen can come back and lay an egg. So when she does these small short moments, like just put the head for less than 3 seconds, we call it a cell inspection. So these are basically cell inspection and cell cleaning behaviors. So I'll talk about colony, the challenges that bees are going on today, and you might all have heard about this colony collapse disorder. It's still a mysterious thing. We don't have a good answer for that, but I'll talk a little bit about this as well. So this is not just one thing. This is just a part of the problem. There are several other problems beekeepers have been going, or the beekeeping industry has been going through. The Varroa mite, which came in 1987 around that time. And that has been a big problem because our bees are not used to Varroa mite. They have not co-evolved with that mite. So they were not able to take the pressure from Varroa mites, and then we had a huge declines. And that's one reason I said in the feral or in the wild, you have only 1% population that is surviving maybe, because they can't take care of these Varroa mites. They have to be managed and somehow treated for mites so that they can be okay. So I'll talk a little bit about colony collapse disorder that you might have already seen in the media and the news. So basically when you talk to a beekeeper, if they complain about this colony collapse disorder, these are the three things they'll mention. Absence of adult bees in the hive with no dead bees inside or outside. So if there is another problem, if there is a pesticide kill, or if there is a problem with a disease or parasite something, then you'll see some dead bees on the bottom so there is a bottom board, and then you see 2 boxes on top in general. So if there is a problem, you'll see some dead bees on the ground or maybe inside the bottom board. And bees are pretty good. They have these bees called undertaker bees. Once a bee is dead, they just take them out and throw them at a distance as well. So they're pretty clean individuals. They'll not even leave a small chip, a wood chip or something. It will be out in maybe a matter of 15-20 minutes sometimes. So they're pretty clean individuals. So this is one thing which the beekeepers and even this community, the scientific community, has been surprised about is there are no dead bees inside. That means the bees have gone for foraging, maybe 2,000 bees in a day, and then they didn't come back. Maybe there was a problem, they couldn't come back. So that's why there was a slow depopulation of the hive, and then we have this problem that you don't see any bees dead inside the hive. And there is a lot of capped brood. You see this picture here, I don't know if it's that clear. So this is all capped. That means there is pupa inside. So after 5 days old, the larva, after they're 5 days old, then the bees come up and then they make this wax coating on top. So they seal the cells, and then the pupal stage starts inside. So they start spinning the cocoon, and then you get a pupal stage. So the bees will never leave this area like this, because they have to have at a certain temperature. It's 33 degrees Celsius or 90 degrees Fahrenheit. That's the temperature inside the hive. So they have to maintain this brood or the young babies that are inside here at that temperature. So the bees have to cover it all the time. So they put their thorax onto that, and then they generate heat so that they are maintaining a certain temperature inside a hive. But if you see something like this, that means the bees have died maybe in the last couple of days or maybe in last few days, so that they couldn't maintain, because the queen was laying well, there is all this brood you see, but there are not enough bees to take care of this brood. So that's another symptom. We see a lot of capped brood without the adult bees. And there is presence of enough food stores. That means there is enough food, the honey and pollen that is inside the hive. So that indicates that they didn't die because of starvation. So there was some other reason why. So these are three different things which we see in colonies that are reported to be dead because of colony collapse disorder. So USDA surveys have been coming up since last 5 years, and the last survey they did in April 2010 reported a loss of about 33.8%. So I'm not saying this is colony collapse disorder. This is a total loss of the hives. So colony collapse disorder may be a part of that. And then losing, for those who are not into beekeeping, losing 10 to 15% is kind of an acceptable number for beekeepers. If you ask a beekeeper who has been into bees for 30 years, they might lose 10 or 15%. Even I lose my experimental hives like 10 or 15% every year. That's not a big deal, because there are several reasons for that. Starvation, queen problems, there are several things. But losing at 35 or 33% is kind of, which is unsustainable. That's a scary number. And if you are losing at that pace, after 2 years, even if you are a beekeeper for 20 years, it doesn't matter. You should be out of business soon. So that's what was making a little scare, and that's the reason why media was after that as well. So today, if you ask me what's going on with colony collapse disorder, so as I said, we don't have a single answer at this point. I'll talk about more in the next slide. So that's why I said there is no smoking gun yet. And there are some bullet holes that people have looked at some correlations in these dead hives. They have sampled these dead hives and found out different viruses and other problems as well. So we'll talk about that in the next. So I'll just mention about this potential factors. When this CCD thing started, so people were having all kinds of theories, from rapture to phone theories as well, which have been discarded at this point. I put some cell phones to show you that they're not bothering the bees actually. So looks like they like the cell phones as well. And the reason why this was discarded is because bees use sun compass orientation for getting back to their hives. You can't bother them with any kind of radiation or the signal. So that was a German study, and he wasn't even looking for anything like this. There was a different study, and then media was so hungry for news, they just picked that news up, and it was all over the media in 2007 that cell phones might be killing the bees as well. So for a single line answer, so these are all the factors that I mentioned here are kind of acting as a perfect storm. So these factors are stressing the bees basically. So when they are stressing the bees, and they are compromising the immune system of the bee. And when your immune system is compromised, then it's easy for an individual or an insect to die because of the existing problems of Varroa mite or the viruses or several other problems which I'll talk about in the next slides. So I've just listed parasites and diseases. I'll go over each of these maybe until here. So parasites and diseases, we talked about Varroa mite. I have still some slides to talk about that. Then pesticides and fungicides, which are being sprayed on the crops. And bees, they don't know that these are being sprayed. So they get the pollen and nectar from those contaminated sources as well. And then there are in-hive chemicals. These are the chemicals that the beekeepers use to kill the mites in the hive. So that can be toxic as well to the bees. And then malnutrition, as we know, we are more into, like, you go to California, we were there in February, and you have thousands of acres of almonds in bloom. And at that point, the bees are getting only one source of pollen, that is almonds. 90% of the pollen that is coming into the hive is almonds, nothing else. So that's kind of a monocropping I'm talking about. So if you have one monocropping, so you're restricting the diet of the honeybee. It's not the same what they used to get 20 years ago. So that's one problem, and then we are having this destruction of habitat, loss of habitat as well. So that's another problem which leads to malnutrition. Then migration, this might be, we haven't quantified migration yet. So when you're moving bees from Oregon to California is one thing, but then bees come from Maine and Florida as well to California for pollinating almonds. And then you're moving, what, 3 time zones away? So as Nick was mentioning, I'm from India, and I visit India every 4 years, and when I come back, I'm messed up for 2 days for sure, because it's morning there in India now. It's maybe 6 o'clock in the morning. So the insects do have biological clock, and we haven't quantified that. They might be going through some physiological stress as well when you're moving bees from, like in spring here, in winter, the bees are in December here, they are in the mode like sleeping mode, I mean hibernation or overwintering phase. And then we move these bees and put in California in January, towards the end of January, when the temperatures are 65 degrees or maybe 60 degrees, close to that. And they find, oh, we are in paradise again, let's forage, and there is nothing else to forage there. And they have to wait until the almonds are in bloom there. So that's a problem as well. And then lack in genetic diversity, as one of our quiz questions were, are honeybees native to North America? They are not. We bought honeybees in 1600s from Europe, or the European settlers bought that, and the Native Americans used to call this white man's fly. They didn't see honeybees before that. So that was a new individual for them. So after that, we passed a law in 1922, it's called honeybee bee law in the United States, where we didn't allow any other species or subspecies of bees into the United States because we were scared of the diseases and pests that can come along with the importations. So that has caused a little genetic bottleneck as well. So that's another, so we have a limited genetic diversity. And there are only, recently there was a study from WSU that they showed most of the queens that are being bought in the United States by beekeepers, they're coming from just like 463 mother queens. So that also tells us how narrow the genetic diversity is in the United States at this point. So these are several factors. I'm not saying this one or this one is killing the bees. These are all different factors. At least the consensus is there at this time. All these factors are playing a perfect storm role and compromising the immune system of the bee. And once your immune system is down, then the existing pests and pathogens can kill the hive. So they are more virulent now. So that's the bottom line on that. So this is Varroa mite, which we talked about, which is #1 pest, I can say still. It's a big threat for the beekeeping industry even now, and that's a major one which beekeepers are always concerned about. So these are the mites which are on the pupa here. So these can cause, not only they suck, this is an ectoparasite, so it lives on the bee and then reproduces inside the cells when the larva is capped. So these are not only a problem by sucking the blood of the bee, but they also transmit a couple of different viruses. So that's the major thing they do. So I have some more pictures here, you see. So this larva is like totally plugged with 7 or 8 here. So this has no chance to even survive after some time. So this is a female here. Males are a little smaller. Usually we don't see them when we are there. Most of the time we see are the females because these die after mating. So this is another symptom. If you look closely here, some of them, they don't have wings here. This is a deformed wing, a half wing, and this one doesn't have a wing at all on the front. And so this is called a deformed wing virus. So this virus is, the Varroa mites are the carriers for that. So the Varroa mites, they vector this deformed wing virus, and so there are also several other viruses as well. So that's the point I wanted to make here, is they not only are sucking the blood of the bee, but they are also transmitting different viruses that are lethal to the bees. And this is another one which was a recent concern, a different species, Nosema apis. So it's a gut parasite which lives in the gut of the honeybee, the stomach of the honeybee, and they cause dysentery in the bees. So you can see the protein metabolism is disrupted. You see all this yellow fecal matter outside the hive. And I wanted to mention that bees don't defecate inside the hive. They have to come out. They can store for a long time in the winter, and whenever there is a sunny day, they come out to defecate. So if you're wearing a white shirt and standing near the hives, you'll be all yellow after some time if you're near that hive. So you see all this yellow fecal stuff here. This is all the fecal stuff from the honeybees, and this might have Nosema apis or serrana as well. So this is another parasite which is a problem, which is a microsporidian. And then we have tracheal mites. It was once a big problem. These days we consider it as a minor pest, but along with Varroa mites, if you see tracheal mites as well. So they live in these breathing tubes of the honeybees. So the honeybees have breathing tubes all over their body on the sides. So if you dissect the trachea, if you see these white small structures here inside, these are the tracheal mites. So they can sometimes clog the entire tracheal tube, and they feed on this tracheal tube as well. So that's another problem we have in the bees as well. And then I talked about in-hive chemicals that beekeepers use to treat for mites, to kill the mites and keep them under control. So these are two different things which many of the beekeepers have stopped using because the mites have got resistance to these two chemicals at this point. Still some people use this coumaphos, which is still working in some instances, but this is totally discarded at this point. So these are two different miticides that are also used, which can stay, and wax is lipophilic. So you put all these things in, the wax can absorb these and they can stay in the wax comb for a long time. And in the long run, they can be toxic to the bees as well. So that's another concern which we have with the in-hive. And then pesticides, as we know, has been in the news for the last couple of years as well. There are beekeepers very much skeptical that pesticides should be taken care of, and I think they have a valid point, because some of the pesticides can be very toxic to honeybees when they are having this additive or synergistic effect. So this is a Penn State study where they looked at all these hive samples across 23 different states, and they found about 121 different pesticides and their byproducts inside the hive. So that's kind of a big, I'm not saying these are all toxic or they're killing the bees, but this is just a broad survey. They did a meta-analysis kind of looked at what they can find in these hives, and they found about 121 different pesticides and their byproducts. So there are studies going on looking at how these different chemicals might be interacting. So I'm talking about this additive and synergistic effects. Maybe a single pesticide in certain dose might not be toxic to the bee, but then you have several, 5 or 6 at least, there are 121 here, but even on average, they may be getting 5 to 10 different chemicals inside the hive. And if they all get like a cocktail of pesticides, then they may have a cocktail effect as well. So that's a big concern as well with regards to pesticides. And if you have heard of this neonicotinoids, this is another group of insecticides that has been very much famous since last 5 or 10 years now. They're extensively used. Even some of them, I don't remember the trade names, but they are used for, like you can spray in your back in your plants as well. So these neonicotinoids can be persistent, and then they have this, they activate these cholinergic receptors, and they hyperexcite the bees or any kind of insect, and they lead to the death as well. So, and some studies in Europe have shown that they can interfere with the orientation for the bees as well. So this is a prime suspect at this point, as neonicotinoids may have a bigger role in honeybee declines as well. But just by themselves not. I'm just talking about they can be a stress factor as well. I know you can't read from the back. I've just listed some. You might not be that interested, but if you remember any of these brand names in this, I've just listed them as highly toxic or toxic. So if you find anything that you are using, or maybe just be cautious when you use these kind of different chemicals. These can be very toxic to honeybees. And I have just a list again here, chlorpyrifos, and these were different pesticides from that study, and there are some miticides they found in the wax, the fluvalinate, coumaphos, and imitraz as well. And then modes of exposure. We always think when we are spraying, we think the bees are only exposed to the direct spray. But when there is a plane coming in almond orchards, they are spraying, and then the bees may be directly in contact. That's one way, and it's a primary way as bee. But then exposure will be also from contaminated pollen and nectar, because these guys don't know which one is contaminated and which one is not. They may go and get those pollen and nectar from the sprayed crops as well. So that's another mode of exposure. And then there is guttation. Have you heard of guttation before? So you see the plants. I think I should show you the picture before I talk about it. you see this corn, I don't know, maybe it's not a corn, but you see this plant, you see these drops. These are not dew drops. These are called guttation drops. So sometimes in some plants, especially in corn, what happens is that there is a lot of pressure from the root when they absorb the water, so the water has to get out some way. And then these hydrothodes of the stomata they have here, so the water oozes from there. So it's not dew, it's coming out because of the root pressure onto the plant. So this mechanism is called guttation in plants. So let me go back. So guttation can be a problem, I'll talk in the next slide. And then honeybees for treated insects, like we have aphids like Christmas tree farmer. If you are a Christmas tree farmer and you think you might not be worried much by spraying, because why would honeybees come to Christmas trees, right? You may not think honeybees are being affected by. But what happens is honeybees have, the Christmas trees have these aphids which sucks the sap, and at least the Christmas tree growers should be spraying maybe 1 or 2 times, I guess. And when honeybees don't have anything else to forage, maybe in July or, in August or September, so they may go to these honeydew, because the aphids, they secrete honeydew. The secretions of aphids are very sweet. We call it honeydew. And the bees will not find any other forage, so they might be attracted to these. And we have seen they go there, and if it has been sprayed for aphids, then they get these pesticides as well to the hive. So that's another way. We don't usually think how a Christmas tree spraying can affect honeybees, but it can as well. And this is an important point I want to make here, is honeybees have fewer genes for innate immunity, hence they are more susceptible. So they have less genes for immune system to maintain their immunity, less than mosquitoes and even several other insects. So the reason why honeybees have less genes is, you can speculate, maybe honeybees have evolved with plants. Plants want these bees to forage on them and pollinate, right? That's the reason the plants have no reason to resist the plant. So there are, like, aphids are ones which suck the plant sap. So they have to produce some chemical to kill the aphids. But they have to invite honeybees because they're important as pollinators. So what happens is, when in this evolution process, we think honeybees have evolved with plants so that the plants want them, so they never were able to develop, they didn't need that many genes for their immunity because they're not exposed to that many toxins there. So that's one theory. They haven't yet proved that whether that's true, but the honeybee genome was out in 2006, and it has shown that they have fewer genes for immunity. So that means they have a very susceptible immune system when compared to other insects. So we have to be very careful when dealing with all these pesticides and different things. So honeybee is a different organism than other individuals. Nick, do I have still time? So this is another study which was done in Italy. So when you think about all these pesticides, so the EPA will give permission for registration because it's not expressed in toxic quantities in nectar or pollen. But when they looked at these guttation drops, the same chemicals, they were expressed in parts per million. So parts per billion is good, parts per million is still worse. So when they used these drops and they collected in a small vial and they fed to the bees in cages, they were dead in like 3 to 5 minutes. So it's different when they are expressed in nectar or pollen, the toxicity is not that much. But when they are coming directly out of these, so this was a pretty new concept that no one was thinking about, that plants can do, like, they know the guttation is a process, but we didn't know that the toxin can come at a lethal dose for bees as well. So, and bees forage for water as well, if you don't know. Bees need water as well, along with nectar and pollen, and especially in summer when it's hot, they have to do this evaporative cooling in the hive to maintain 90 degrees temperature. So they have to collect water as well, and then that can be toxic to them as well. So in next 10 minutes, maybe I'll try to just wind up with the research priorities that we have. I still have like 10 minutes? Okay. Yeah, I think I took too much time on explaining the other things. So these are 3 different areas which we are focusing on at Oregon State: honeybee health, honeybee nutrition, and honeybee pollination. So I'll just go over honeybee health. So what we are doing here is a comprehensive analysis of honeybee health. So I have been here in Oregon State for only 2 years now. So since we came here, I'm looking at this pest and disease complex. So we have randomly sampling beehives in the state of Oregon, looking for Varroa, Nosema, and Tracheal mites. And then we are doing a nutritional analysis. I mean, we take these glands off from the head of the honeybee, and then we do a protein analysis to find out the protein quantities in each of these bees. So that's a good indicator for the nutritional status of the bee. And then we are also looking at pesticide residues in the comb. It's still pending. We still have we're looking for funding yet to do this part. So it's an expensive thing to do pesticide analysis. So those samples are still waiting in the freezer for that. But these two things we are regularly doing every year to look at a big picture of what's going in Oregon. So we send these sample kits to the beekeepers, and then there is a set of questions. You can't read this, but I'll read one or two from here. What's the current size of the hive? What treatments did you use for controlling Varroa, Nosema, and Tracheal mites? Did you provide protein supplement? Was this colony established from a swarm? How old is the queen? Those kind of questions, so that we can go back and correlate the hive loss to all these things. So here we have an example. I have a wonderful people helping me. This is Carolyn, you see, sitting here as well. And we have been getting about 750 we distributed. Maybe we got like 400 back, I guess, until now. And we have a website where we put all these results here. So you have the sample numbers here, then Varroa mites. So if someone sends a sample and he has 290 bees, so we find, okay, 0 mites or 5 mites, 313 bees, and then we'll put these results for tracheal mites, whether absent, present, and we are giving a score as well for those. And then this is Nosema spores per bee. So this is a parasite which I showed some slides before. So we look at those things, and then protein content for it. So these are not only useful for us to get a big picture of honeybee health in Oregon, but the beekeepers can go back as well and look, if you're a beekeeper, you send me this sample, so you know what exactly is in their hive, and then they go for treatments based on that. Or else in April, we are also collecting the status. So if the hive is dead, if they report that the hive is dead, that they sent the sample in August, then we can go back and see what was wrong, what might be responsible for those problems. So there are different ways to look back and see how we can interpret the data from there. So I'm just giving a brief picture. We are doing some more things, but I don't have time to explain all, so I'm just touching a little bit on each of these studies. So then other one is honeybee nutrition, where we are looking at the nutrition, because I think nutrition is the first line of defense, because as humans, if we are consuming chicken or maybe just one kind of meat for maybe 2 months, your immune system is gone. So it's similar to that. So we are looking at different things that might be affecting nutrition in the bees, and looking at how it can be a first line of defense for these guys. So we are looking at different, and there is one study already that has shown that nutritional stress or poor nutrition is one factor why we are losing colonies. So this study correlated the vegetation available and the number of hives that died in that state. So there was a kind of a positive relation or a correlation between the nutritional status and the colony losses. So we are focusing on nutrition to see how it's going to affect the colony losses or the colony immune system. And as you saw in the quiz, the primary source of protein is pollen for the bees. And it's not only protein, they get lipids, minerals, vitamins, and several other things as well. So this is the protein cycle. So you see a pollen forager, she collects the pollen, puts in the hive, and the nurse bees are the ones. Not all bees eat pollen that they collect. So the bee which is collecting the pollen, she doesn't eat it actually. She goes and puts it in the cell, and the specialized bees called nurse bees are the ones which are going to eat that pollen. Then they grow these glands in the head, and they produce this white secretion which worker jelly we call, and bees provide it to the larva for the growth and development. So that's how pollen is being recycled inside the hive. So in this study, we are looking at pollen quality effects on honeybee physiology, colony growth, and immunity or immunocompetence. And as I mentioned, monoculture and loss of habitat are the ones which are restricting the diet. So we chose almond pollen because this is one big crop where beekeepers go and pollinate the crop, and the bees are there for a month and a half or 2, and they are relying on just one source of pollen there. So this is a single source, and this one has at least 5 or 6 different types of pollen. So we have a flight cage here on Peoria Road. If you're interested, you can come and look at our research as well. So we have these small hives here, nucleus hives, where we put these pollen. So the reason why they are in the cages is we don't want them to get their own pollen from outside. They have to rely on whatever pollen we provide them, so that we can compare the differences between feeding on a single source pollen versus a multiple source pollen. So this is just a design, and you provide water, everything inside the hive, and these are the things we are measuring inside. So the brood area for 6 weeks, hypopharyngeal gland, protein content, lipids, sucrose response thresholds, and immunocompetence. So you put the pollen, you grind the pollen, put it in the cells like that, and then spray with a 30% sucrose solution. so that it becomes attractive and live in the hive. And the next day if you go, they might have packed it well like this. And then this goes back into the hive again, and they start feeding on this pollen. And we do a Bradford analysis, and we also measure the size of the hive. So we are not literally counting the bees here, but you can see this is a square inch grid. So there might be 8 to 9 bees that can fit in there. So you just count all these number of squares there, and then you interpret how many bees are there in the hive. So we look for the brood and all those small details inside the hive. And there is another way to look at the behavior as well. We haven't done this yet, but I'm just giving you some examples which I did when I was at Texas A&M. So there is a big association or a robust association between sucrose response thresholds. I know it might be a confusing term here, and I don't want to confuse you anymore, so I'm not going into that. I'll just show you the next slide. But yeah, if you want, you're curious, you can talk to me after this. So there is so each bee has a different sucrose response threshold, just remember that. And then that can affect the foraging behavior of the bees as well. So to look at that, if nutrition is affecting their sucrose response thresholds, and maybe it might affect the foraging behavior, we do this thing called proboscis extension response. So we put these bees inside these holders like this, and then we test them with different concentrations of sucrose in logarithmic scale. So you touch the antenna. So their antenna are like our nose. They can smell through their antenna the concentration of the sugar. So you touch the antenna and then see if she's sticking the tongue out. So can we start this thing? So this is a bee here in the, yeah, it looks funny for us, but for not the bee, I guess, because, but you can release them. Yeah, we are not killing that bee actually. She can be released and she can go back home after that. So you touch the antenna, and then you'll see she's sticking her tongue out now. So we don't provide the drop because it might confound the next set of sugar that I'm giving. So you are just kind of teasing them. You're not providing the sugar at that point. So this is the next set of concentration, and we see whether she responds to the next concentration as well. So we look at, if she responds, you put a plus. If she doesn't respond to that concentration, you get a minus, and then we can arrive at the sucrose response thresholds from that score. So that's one way to look at, and you can train bees as well, like dogs. You see all this Pavlovian technique where you ring the bell and the dog comes and salivates for food. So the bees, some people have done some of the memory studies you can do. You can send a coffee smell. You can have a fan here and put your coffee here and then pass the air through that. And then once the smell goes onto their antenna, give them a drop of sugar. And then you can go back after some time, you do 3-4 times, and then you just send the smell and they'll stick their tongue out. They think that sugar is gonna come after that. So there are ways to do that, and that's a nice way to look at. Some of studies have shown that they have looked at if there is a memory loss in bees. So if your bee is affected with some kind of an insecticide, and if there is a memory loss or orientation problem, so once you have 2 sets of bees, this one was contaminated with pesticides and this one is okay. And then you pass these and see, these guys will not remember after 10 minutes. If after 10 minutes, you give a time break of 10 minutes and then blow this coffee smell again on their antenna, they'll not stick their tongue out because they have lost their memory. But basically the ones which are healthy, after 10 minutes you pass that, they'll be still sticking their tongue out. So there are ways to test the memory loss as well in bees.
How do you get the bees in there?
Good question. That's a secret. You have to pay me for that. So. No, actually, it's not that hard. You can, we sometimes knock them down with CO2, carbon dioxide. You can put in jars and then release carbon dioxide for maybe 30 seconds, and they just are flat for some time. Then you put them back and they are alive again. So that's one way. But sometimes we get stings as well if we are doing alive. So that's fine. We live with that. So. And we are looking at this immunocompetence as well. That means the immunity of the bee. So we take 10 microliters of the bee. So we bleed the bee. Here the bee is not going to survive. We are killing the bee basically. So you bleed the bee and take 10 microliters of the blood and then look for these different enzymes. So these are good indicators for the immune system of the bee. So that's another thing we are looking at as well. So I don't know if you might be interested in the results, but I'll just go pretty quickly on this. So we looked at the mean hypopharyngeal gland protein content and we saw the multi-source pollen were significantly higher. We were expecting that actually, so it's not a big surprise. And then the mean brood area, at least from week 5, we see a significant difference here. So the multi-source pollen is still doing well as far as colony growth. And then this was a big surprise. We didn't do too many reps, but whatever I had, you see the Phenoloxidase, this is one enzyme here, and this is the multi-source, the black ones. So you see the ones with multi-source pollen feeding had the best immune system, and these were pretty low actually. So that tells us that the immune system can be compromised by just feeding on one source. So that's what I have put here. And this compromised immune system may render the bees more susceptible to existing pests and pathogens. So in future studies, we want to see if we can do the same thing feeding with different sources, and then also put them with or infest them with different problems like the nosema or other problems that we have, and see if they can survive better in case of multiple source pollen versus a single source pollen. That's another way to look at. So I think I have 2 more slides to go. So, and I just wanted to tell you that application of pesticides and protection of bees are not mutually exclusive. That I mean that I understand that farmers have to protect their crops by spraying for insecticides to kill the pests that are affecting their crops. But then beekeepers also have to keep their bees healthy and not contaminated with all these pesticides. So it's kind of a tricky balance that they have to. So that's why I put like there is a beekeeper-grower cooperation is very much needed, because without that, it's impossible to keep both bees alive and also the crops. So that's what when I talk to growers group, I try to emphasize that, and most of them try to understand that it's important, and especially if they're hiring bees for pollination, they pretty much understand. But if you have something like Christmas tree, maybe you may think, I'm not killing the bees, why should I be worried about that? But I think it's a valid thing that you have to worry about bees as well, because they are pollinating several other crops as well. So using all pesticides, these are small things, many people know all these, but just a reminder that all pesticides should be used in manner consistent with the label, and then avoid insecticides with long residual hazard. You can find this list of toxicity levels, how long they can stay on the crop. So you have to be very much aware of that as well. And how growers can help, again, some points, avoid spraying during bloom period. Many of them try to do that, but sometimes it's not possible, I guess. And if possible, spray in the evening or early morning, because the bees are mostly home at that time, early morning or late in the evening. So if possible, spray should be done at that time. And then controlling blooming weeds in crops. So we think, okay, weeds are okay, and we spray the crop. But then if the crop is sprayed, and then there are blooming, there is these blooming weeds you are spraying, and then they may go onto the weeds as well. So they have to kill the weeds as well before they apply these pesticides. And this is another thing which I always mention is provide habitat for bees, planting hedgerows. So you might have heard of this concept for maintaining diversity of pollen and nectar for bees. And maybe this is valid for most of you, that what you can do to help bee pollinators. I'm not talking about honeybees, all bees, native pollinators, insect pollinators. So plant and encourage. They have listed some crops, some plants here: Phacelia, red clover, foxglove, basil, rosemary, bee balm, lavender is also attractive to bees, goldenrod. So these things you can plant either in your farms or yards or gardens, whatever you have. And those things can be a continuous source for bees when there is nothing else available for them outside. So that's one thing I would encourage you to do, and appreciate the services. I think you are here, that itself means that you appreciate the services of bees, and then I want you to create awareness about their importance in your community as well. And if possible, become a hobby beekeeper. I promise you it's the most fascinating, most entertaining, and most satisfying hobby, I guess. So try to become a beekeeper if you have a place to keep bees. And I think I'll take questions and see if I can answer it. Thank you.
The person with the honey, the pollen.
Yeah. And now I think you guys can taste some honey.